Hello everyone and welcome to today's Physics Daily Booster. This is number 5 in our Paper 2 series, and while it's not explicitly listed on the advanced information for either the foundation or the higher physics, as we've found out from our previous ones, then that doesn't mean it's not going to come up. So still worth a watch because there will be potentially marks on this on your paper. What we're going to be doing in this booster is having a look at lenses. So first thing, our first type of lens is a convex lens. Now, convex lenses are sometimes called converging lenses, and what they're going to do is refract those rays of light that enter it to what we call a principal focus or a focal point. Now, this is the kind of lens that we're going to use in magnifying devices. So if we have a little look at the diagram just underneath here, what we can see is these little red lines coming in from left to right, those are our rays of light. Now do notice that across the middle there we have one labelled the principal axis. Now if you notice, the principal axis is literally a horizontal line that runs straight through the entire lens and beyond. So principal axis will usually be on the exam paper itself, along with the diagram of the lens outline. What you are likely to be asked to do is something with the refracted lenses. Now, what we actually find in our convex lens is if you find that midpoint of the lens, so this little dotted line running down the centre here, then what you'll notice is those rays of light come straight in and they are literally parallel to one another until we get to that dotted line running through the centre of the lens. At that point, they then refract, so they bend. What you'll notice is the two above the principal axis are going to bend down, and the two below the principal axis bend up. Now the key thing is this principal focus, this little point here, is where all of those rays actually meet, so they converge on that one point. And the distance from that dotted line in the middle of the lens to the principal focus is what we refer to as the focal length. The little dotty line that we've called there, that is also our optical centre. You'll notice in the very middle here is our little marker for the optical centre. Lens number 2, concave lens, also known as a diverging lens. This is going to spread light out. So convex brings them together, concave sends them further away from one another. So we still have our principal axis that runs straight through the optical centre there. We've still got our rays of light coming in parallel to that initially, but we then get to obviously that central point, our little dotted line there through the lens, and when the rays of light hit that, they bend away. So notice how they're starting to move up at the top and down at the bottom, so they're moving away. If we want to know where the focus is, it's called a virtual focus this time, because we then have to track those rays back. You can see the little dotted lines on the diagram to a point where they all meet, and that will be before the actual lens itself. So principal axis will have this little point where all of those rays of light meet when we extend them back, and that is our virtual focus. Focal length in this case is between the optical centre and the virtual focus. So why do we have these types of lenses then? Well, one of the primary ones is because we're going to use them to correct different vision defects. This is something we've talked about in biology already. If you are short-sighted, that's where distant objects appear blurry, and the reason for that is that the rays of light are actually focusing in front of the retina, so it gives you that blurred image. In order to correct that, what we do is we use a concave lens, so that spreads the rays out, and then what they're going to do is as they pass through your lens, that will obviously refract them inwards and they now focus on the retina itself. The opposite to that, if you're long-sighted, then near objects appear blurry, and that's because the rays are trying to focus behind the retina. So what we do in that case is include a convex lens in your glasses or contact lenses, and what they're going to do is start bending those rays of light in before they pass through the lens of your eye. So that means we again get those rays focused on the retina. So make sure you remember short sight, concave, long sight, convex. They can ask you to draw ray diagrams including lenses. And what we've got are kind of 3 key points, if you like, to help you to do this. They will give you part of this printed on the exam paper. So what they'll probably do is give you the principal axis, the object and the lens itself on the actual exam paper. You then have to add the other rays. So if we look, the first thing you're going to do is draw that red line. If we look at this top diagram here. So you draw the red line from the top of the object to the lens, running parallel to that principal axis. So a nice parallel line from the top of the object to the center of the lens there. Once we've done that, what we're going to do is draw a ray that goes from the lens through the focal point. So what we do then is we take that one down through the principal focus, which is going to be here, be marked on your diagram, and you just extend that line further down to where we're uncertain at this point in time. The next thing we do is our blue line, which is literally you place your ruler so you've got it nicely at the top of the object, you run it straight through the very center of our lens right there, and what we're then going to do is just draw one straight line, and what you'll find is there will be a point that those 2 lines that we've just drawn actually meet, and that is going to be the tip of your image. Once you've got that, obviously you can then just fill in the rest of the image itself. In this case, just the arrow, as we can see there. At the bottom, you can see an example of the kind of diagram they could give you. So we've got the object, the principal focus, the lens, and of course the principal axis. All you then have to do is add those rays on there. Don't forget to add the arrows to show the direction at the end. So what do we use lenses for? We've mentioned obviously correcting eyesight, but there are also other uses of lenses we need to be aware of. First of all, our convex lenses. These are the ones we find in magnifying glasses, cameras and projectors. We need to know the type of image that's created by these different devices. If we look at a magnifying glass, it's a virtual image that is magnified. The name kind of gives us a hint on that, and it's upright because when you hold a magnifying glass up, it looks the same way up as you look at it. A camera is a real diminished, so smaller, and it's inverted. And the projector, real, magnified and inverted. So you've just got to learn those. There's no trick to this, it is just sit and learn them, I'm afraid. Concave lenses, 2 examples here, spy holes and the rear windows on coaches. But the good news is the type of image is identical for both. They are virtual, diminished and upright images. The last bit that we're going to look at in our booster today is colour. You should have done this experiment at some point in your time at school, where you get your little ray box, you shine light through a glass prism or a plastic prism, and then you get a nice little rainbow on the other side. Now, what we're actually doing is we're splitting the white light into its individual colours. Now, this is in a process called dispersion. Make sure you do know the correct sequence of our colours, so red, orange, yellow, green, blue, indigo, violet, because they could ask you to fill those in. Now, the reason that this happens when we actually shine the white light through the prism is because each of those different colours has its own frequency. Different frequencies of light travel at different speeds through glass. So as a result, each of those individual frequencies of light is going to be refracted by a different amount. The higher the frequency, the greater the degree of refraction here. If we then think about one other factor to do with colour, we need to understand the retina. And again, this is an overlap with the biology work we did in B3. So your retina is actually sensitive to red, green and blue frequencies of light. So you've got cells in your eyes, if you remember they're called the cone cells, that are able to detect those 3 frequencies. So what happens is those receptor cells detect the frequencies, send signals to your brain, and then your brain interprets those signals and gives you this perceived colour. So it basically tells you what it is you're looking at. The next little bit is looking at filters. when we talk about a filter, this is something that we can add to a light source that's going to absorb a range of frequencies but leave some. Now, if we look at our nice little example here, we're starting off with our white light. I've drawn all the individual colours just so we can see what's happening more clearly. So red, orange, yellow, green, blue, indigo, violet, that is just white light coming in. In this case, I put a red filter in. Because we have a red filter, that will absorb all frequencies of light except for red. So filters absorb all frequencies of light except that of the colour that they are. So remember that phrase there, absorb all frequencies of light, except obviously the colour it's made from. So as a result, the light coming out the other side of the filter is red, because the red light is transmitted. If we were to add a second filter in here, so if I was to add a blue filter, I know it's drawn in red, but imagine. So if I was to add a blue filter at this point, the red light that went through our red filter would now be absorbed, and therefore no light would actually pass through. Watch out for different ways they phrase this. So sometimes they say something along the lines of, you have a blue book under red light, what colour does it look? Because you've only got red light, that's only the red frequency. If it's a blue book, it will only transmit the blue light. There is none, so it appears black. So basically what you're looking for is any colour that doesn't match with your filter or the colour of light, then it will be black. As always, if there's bits you'd like to look at in a bit more detail, you can use the P5 playlist on the channel to help you out, your revision guides, your classwork notes, and as always I recommend having a look at some of those OCR exam papers to get a bit of a feel for the kind of questions that can come up. And of course, don't forget to join us again tomorrow for our next Daily Booster for Physics.